We are very excited to have you here to our working group on light, which is a very interesting topic which should interest everyone because it affects all of us. And if nothing else, hopefully today through a series of really great speakers, we will explore this a little bit. So my name is Elke Buschbeck. I'm a professor in biological sciences. I'm also this is a neuroscience graduate program because neuroscience is one of my backgrounds and interests. And most of my life I've been chasing the weirdest animals, well, amongst the weirdest. The others are very weird. Full disclosure, but anyway. But more recently I've also been interested in light.
Yeah, I'm Dr. Chris Holland from the School of Arts, and I'm interested in art history and how light has been portrayed by artists in the past and in the future mostly. So that's what we'll be talking about this afternoon. So please stay after lunch and come to the arts, architecture, design portion of the program. So I'm excited to be working with Elke on this light project.
Yeah, so for the working group, the overarching goal is a book. And there's already one publisher, Springer, that is very interested in it. The idea is that the book is going to be generally understandable to the broad public that's scientifically literate. The hope is to get a lot of awareness from people about, the enormous importance of light in our lives, because I think a lot of that is lacking, as we also will explore today. For the working group, we have lined up speakers, so there are programs online and flying around. There are two sessions. We're about to kick off the first session, which is more like science geared, and then in the afternoon, so then there's a discussion, and then in the afternoon, we'll have a more art architecture geared session. And so those people who are talking, we right now look at them as the chapter leads, and we hope that if you're talking, you're willing to take this on. But in no way the book has to be following, exactly those type of topics. So we're hoping we have this weekend for a lot of crosstalk and for a lot of brainstorming. What really should be in the book? What actually makes it complete? Are we missing anything? Who wants to actually maybe co-author, even among the chapter leads, to tackle a certain topic, maybe even a topic that's not on the list yet. Like I can think one future-looking one that might be interesting to add. And then we have, but actually very briefly, can you raise your hand if you're a chapter lead? Also one of the speakers today, just so everyone, that's an idea. Then we invited 13 people who applied to the working group. A lot of those are students or also who moved recently to Cincinnati. And they're invited back to come tomorrow. So today is open to the public and this is being live streamed. Tomorrow is the inward-facing working session where we hope to really explore this further. So those of you who are the other attendants, like the applied, admitted attendants, can you just raise your hand? Okay, cool. And then we have a few people who are in the general public, and then in cyber world, I have no idea in inside. So, but again, this is recorded too, and also can be looked at later. So that's the main.
Yeah, I just want to reiterate the goal of this morning is to, after the presentations, have a chance to break out. So you notice the boards behind you. The people presenting will be at their boards, and you can give feedback and ask questions at that point. Because the presentations are stacked back to back, each 10 to 15 minutes. So that's sort of the general idea for the morning.
Actually, presentation we have 15 minutes to present plus 5 minutes for
a quick Q&A. Yeah,
a little bit of a discussion.
Great.
Anything else at this point?
I think we're good. We can field questions along the way if people have them.
Yeah.
But I think the general spirit is to begin a cross-pollination between science and the arts and create a book that reflects that. So that'll be really interesting, I think, to both the general public and hopefully yourself. So we're excited to co-collaborate. So yeah, I think.
One last thing. For those of you who are our speakers, just keep in mind this is a very broad audience. And again, the goal is to have something public-facing. So hopefully we all can work on our language. And everyone in the audience, if there's ever any one of us who uses any jargon that's not really clear, please don't hesitate. Like, please can you explain what that is? Because it's so easy to be in the field and use vocabulary and assume that everyone knows it.
Yeah.
And so we want to make sure things are as clear for as many people as possible. All right?
Yeah, I think we're ready for.
Okay. Well, thank you for
There's a microphone too.
thank you for including me. I'm actually excited to see what's going on. And as a photochemist, we're always excited, right? And that's an inside joke. But what I do, I work at UC and I study how light interacts with material or how we change molecules. With light, we do all these chemical reactions. Does this work? No. Well, doesn't matter. Here's another one. We do chemical reactions, and that is we turn one molecule into another one. And I use light for this, and I use light to detect what happens in between, okay? And so we are actually illuminating pathways, we're identifying pathways. We use light to break chemical bonds and form new ones. And this is just like hiking, which is one of my favorite activities. This is my daughter hiking, and she thought I was old and annoying, so she hiked ahead of me, and she's going down to this spot down there, and it takes about 5 hours to get down there. And of course, she made it several hours before me. It's 24 hours daylight, so you're not really worried. There are no trees to get lost. So this is what I do in my living is I look for pathways. So we all know about energy crisis, and I'm not going to go into that. Chemists, they tend to work on coming up with solar energy. How can we actually harness solar energy to do something to solve the energy problems? You look at these things, we know coal and oil and natural gases. This is what we want to move away from, but we need a lot of energy. But chemists don't just work on making cells that will harness. We use a lot of these. We're trying to come up with synthesis that rely only on sunlight or try to do more of it, because the earth is bombarded with sunlight every day, and all the energy that we were bombarded with, this could actually solve all our energy crisis. But we could do more than just solar energy. We can look at reaction. So if you want to synthesize a pharmaceutical, it would be really neat if you could do it using either energy efficient LEDs or just sunlight. Of course, biologists are way ahead of us, the chemists. Nature knows how to do this. You have sunlight and you use all the plants to turn carbon dioxide and water into energy or sugar, and you give out oxygen as well. So this is a way to transform light into chemical energy. And this is what chemists are working on. Just we're a little bit behind nature in that sense. You can do all kind of photochemical reactions using lights. So here you have a starting material, which is provitamin D. It's in your skin. You shine light on, or you expose your skin to sunlight, and you form pre-vitamin D. You're breaking this bond here, and you're forming these double bonds here, and then you just need rearrangement to get vitamin D. So this is how photo reaction happen. The starting material absorbs light, it gets a lot of energy, and this energy of the excited state allows it to form products. but I promise you I won't go into too many chemical reactions. If you think about it, we have a lot of smart material. Smart material is material that respond to heat, light, or any kind of external stimulus. And if you're a biologist, you know that sunflowers, they follow the sun when they are younger. Polymer chemists have been trying to do this, and they do this very effectively. They have what they're called photoresponsive polymer. This is a polymer here. It's actually following the light, right? So you can do things similar to nature, and there are a lot of application for smart material. And okay, the other smart material you're all aware of are your sunglasses. They turn, or you have glasses that are transparent inside and then they turn darker when they see light. These are examples of smart materials. But this is a polymer, right? The polymer is partially crystalline. Crystalline means it's perfectly lined up and repeatable unit. The rest of it is amorphous. So when you play domino like this, where it's lined up, it actually shows a great response. But where it doesn't, the response is less. And this is where we, the chemists that are non-polymer chemists, we thought, can we use crystals? Crystals are always perfect lined up. Everything is lined up. And if you play dominoes where everything is lined up, you'll get an extensive response because everything falls in place, right? So this is an example of where they did a photoreaction. This here, you can see these are molecules that are lined up. They're tethered to this backbone of a different molecule, and they're lined up perfectly. Where you see these, this is where you form a new bond if you shine light on it. When you form the new bond, you can see you have a 4-member ring here instead of just these double bond. And interesting, when this reaction takes place in the solid state, these are the crystals, just normal looking crystals. And if you expose light on it, the crystals will start jumping. They're just putting a UV light on it. And they jump and they fall apart. And this is because of a chemical reaction. Okay? And this is actually turning light into movements. And that is what we have been studying for the last few years. But what we do is we look at reactions that release nitrogen gas or CO2, mainly. So we take compounds that have N3 group, it's called an acido group. You shine light on it, you will release N2 within the crystal lattice. That builds up a lot of pressure. And we think that we're actually cracking the crystals or doing the reaction, but the response of the crystal is due to the nitrogen gas release. So we think we have crystals, we're generating nitrogen gas within them, and that gets the crystals to move. And this is, I'll show you some examples. This is a compound, this one here. It undergoes a reaction in the solid state. This is a crystal of it, and you see then light starts shining on it. You form bubbles within the crystal, and the crystal stays intact. But as we continue shining light on it, it will actually start cracking, right? And we're releasing gas, and the crystal crack. And the interesting thing is the crystal always cracks the same direction. So we can identify in this where the nitrogen gas is coming off first. And this is a compound we, of course, study it. We know what the mechanism is, but I promise I won't tell you. But this is the compound it forms. Then we took a derivative of this compound. Very similar compound. This is now in an oil bath. So you shine light on these crystals, and these crystals, they undergo exactly the same photochemistry, but the crystal responds very differently. You can see you get nitrogen bubbles coming off where there is a defect in the crystals. And as we continue the reaction, there are more and more defects because the product is like a defect. And it comes out, right? And this is exactly the same reaction. So we know it's not the photoreaction, it's the response of the crystal that matters. This is forming the same kind of compound. And this is something we've had fun with. And we are trying to explain by doing calculation on the crystal structure, explaining where there are weak. These are represent strong interaction. These are the weak interaction. These are the two different derivatives. And for the compound that actually stays intact a lot longer, we have stronger weak bonds. And this is something we study in details, but I'm not going to go into that. We thought about it. Since we're not synthetic chemists, my students don't love doing columns, purifying compound. But we actually take these compounds, they form this if you shine light on it. And this is actually a pharmaceutical relevant compounds. And we can make this reaction just by using a green laser pointer. If you take a laser pointer, you shine light on the crystal will actually fracture very systematically. And because it continues fracturing, you expose a new surface, and you can take this to 100% conversion. We had a high school student that sat on the roof for lunchtime with crystals to make this happen, right? 100% conversion, no need to purify the compound afterwards. She decided that chemistry was boring. It was so simple, and she's graduating with a computer science degree from Stanford. But we're very proud to be part of her journey. We can also do these, even though my reactions are irreversible, because nitrogen gas is so stable, we can never trap it again. We can do bending of crystals, so we can do more applications, and that is what we're having fun with, kind of exploring. But then my student decided what is the most important application was to blow up a balloon, proving that we're actually releasing nitrogen gas. But what we didn't know initially, there are no transparent balloons. They're actually transparent condoms, and we just had to wash them before we use them to take the lubricant off, and you can blow it up. It proves the point, right? So now that I've convinced you that I am totally crazy and I spend money on condoms, video cameras, and tripods for my students, I'm going to say thank you for your attention. I'm grateful for NSF to give us money to do these crazy things. And I'm actually grateful for all the students that have worked in my lab and make this possible, and collaborators across the world. And thank you for your attention. Yes, questions?
Great. Behaving crystals, I love it as a biologist because, you know. But what I was going to ask you is you showed us the first one. You said you had UV light, and then you had one that had green light. I was just curious about the different wavelengths of light and how forgiving are each crystal. Do you have really narrow wavelengths? Is there a pattern of which light works and which one doesn't?
So, because we're photochemists, we start by obtaining UV spectra or absorption spectra. So we know if we want to do a gentle reaction, we go as far away from the absorption where we just have a tiny absorption, because then we don't touch the photochemicals. The reactions are a lot cleaner. Because the green light, that guy actually absorbs, it's yellowish, you can use green light for it. Blue light works just as well. We just look at it, where do they absorb, and that tells us where we can do that. Occasionally you get different reactivity. Yes.
What are some of the applications that this chemistry would be like?
This could be like sensors, actuators. I mean, we're playing with different weight. So we take these crystals that can pretty nicely and we do. We're not true engineers. So we try to explain the fundamental, but we're having fun with how we do that.
One last
Questions? Yeah, I'll set up.
So I love that you use these like so photochemistry, I haven't seen that term before, and as someone who does photography, I find that super interesting, maybe a point of synergy for later discussion, but I think some artists would be interested in like moving things and maybe trying to make them that sort of stuff, to bring some more interesting of your lovely group, a couple masters or.
Yes, I mean, anyone, because I mean, we suck at taking these videos. This is something we just learn.
Yeah.
One of my students went out drinking and met a young, nice woman that made music to his crystals, but then they broke up. So but yes, anybody that can help us with making this a little more artistic. But thank you. Let me get out of your way.
Next speaker is Dr. Todd Oakley, joining us from the Department of Evolutionary Biology from UC Santa Barbara.
Seeing if I can get my computer hooked up. It's weird, it's detecting here.
Did you choose
Since
the
laptop.
Yeah.
Yeah, no, you have to choose this one.
Oh, DocCam.
Yeah, that's the only one that works for me.
Okay.
I think maybe it's a projector.
Yes.
Copy it over. Maybe somebody else should go first.
It flashed.
Yeah, it seems like it's detecting here.
No
So
recognition. Why don't you flip over to this?
I could go on the computer too without videos.
Yeah, I think I would try this first.
Okay.
Try that and see if that makes a difference.
Oh, hold on. It just asked for, yeah, allow. Well, go off the drive it up in some of the videos, which should be fine.
So just try this one.
Okay.
This is the only one that works for me.
Oh, yeah.
Is that you?
Oh, that is the laptop, yeah.
Okay, thank you. All right.
Okay, my apologies for that. I have a few videos, so I wanted to use my laptop, but just didn't test it beforehand. So imagine that you are an alien coming to Earth, and what would be the thing you'd notice about the Earth? And I think, first of all, you might notice the water, but I think really the most, of course, remarkable thing about Earth is the riotous diversity of life present on Earth. And Dan McShay talks about 3 hallmarks of life. One is the amazing fit of organisms to their environment. Second is this riotous diversity that I just mentioned. And the third is the intricate complexity of life. And while we have a good explanation for this adaptation since Darwin and of natural selection, what McShay and Brandon argue is that we don't have a very good explanation for how biodiversity and complexity originate. And that is the topic of my research interests, which is how do complex systems originate during evolution? The way that I define complex systems are those with many parts. That's following Dan McShay, who was inspirational for me. And to understand how those complex systems originate, I basically tell origin stories. And origin stories are tales of transformation that define a new pathway in life. And I'm taking that from a book about superhero origins that I've been reading by Rosenberg. And she classifies origin stories into 3 different categories. One, those due to chance. Another, those due to trauma. And the final one are those due to destiny. And I'm going to tell you a little bit about each one of those. So chance, of course, Spider-Man just happened to have gotten bitten by a radioactive spider. Batman witnessed the murder of his parents, which transformed his life pathway into a superhero. And destiny, well, there's a few, but we can think about Buffy the Vampire Slayer, who was born to be a slayer. So I'm going to go through 3 stories related to my research. One about chance, one about trauma, and one about destiny. First of all, chance. So when I was applying to graduate school, I wanted to understand evolutionary history and see if the tools that we use were good enough. So I wrote to my PhD advisor to be Cliff Cunningham, and I asked him if there's a group of organisms with a really good fossil record that we could use and pretend like we didn't know the fossils and see if we could reconstruct the actual history. And the title of his email came back and it said, "Ostrocads are it!" Exclamation point. And I didn't know what an ostrocad was. He accidentally inverted the O and the A. He really meant ostracods, but I also didn't know what an ostracod was. They're these tiny little crustaceans. And I went to the literature. At that time, I had to go to the library physically. It wasn't online yet. And the first paper, by chance, that I found was this one called Discovery of Functional Iridescence and its Coevolution with Eyes in the Phylogeny of Ostracoda. So this paper proposed that eyes evolved separately within this group of ostracods compared to everything else. That set me on my path by chance, thinking about, well, how is that possible? And Andrew Parker argued, these findings challenge current theories which demand a single evolution of the compound eye. That seemed really amazing to me, that a compound eye could evolve separately from all these other compound eyes. I don't believe it anymore, but at the time I did. I thought it could be possible. And that became my PhD thesis, was looking at the evolutionary history of these crustaceans and mapping this trait, presence or absence of eyes, onto the evolutionary history. And that's what I do, is map traits. And if close relatives contain that trait, we think their common ancestor did too. And then we can use mathematical models to map the entire history and look at when these different traits evolved. But what I realized was that this was a vast oversimplification, because we're talking about complex systems. And so this is saying, well, the system is either all here, the eye is all there, and here it's all gone. And so that inspired me to look, instead of it as all one thing, to look at the parts. And that is really my research program, looking at the parts of these complex systems and tracing their history. That leads to trauma. All right, so imagine that we know about trauma, having just been through lockdowns and things like that. But the great thing is that sometimes trauma can lead to new complexity and new things, innovations. So Zoom, for example, think about that, how good we are at Zoom now after being on lockdown. And there are other traumatic things in the environment. So, for example, think about something that reacts with cells, toxic to cells, creates reactive oxygen species, toxic aldehydes. It's pervasive in our environment. It breaks the disulfide bonds of proteins and creates cysteine radicals in these proteins. Even the core of life, it breaks the DNA itself. So what am I talking about? Of course, I'm talking about light. And we don't always think of light as a toxin or as traumatic, but it really is, especially the UV light that contains a lot of energy. And this inspired us to think about this, and we started noticing that all the parts involved in the origin of eyes tend to be related to reactions to photooxidative stress. And so just mentioned this briefly, but we wrote a review on this, suggesting the idea that all these different parts, and this is from Dan's graphics and his inspired by his research and writings, all these parts that are related to the origin of eyes, if we trace their deep evolutionary roots, we think that they're originated first in photooxidative stress responses. So that might get you thinking, well, there's light that hits our body all over the place. Why are there not eyes everywhere? Well, in some ways there kind of are. They're not eyes per se, but there are light responses all around our body. And just mention a little bit of research of things that we found. This is a hydra, a small little anemone-like creature that lives in fresh water. And we started looking for the most distant relatives of humans that have genes called opsins that we use as humans to respond to light. Well, hydra also have opsins, but they don't have any eyes. But the genes are highlighted in purple here, stained in purple, are all around their body. And they use these genes, we think, these are the stinging cells that cnidarians use, like if you get stung by a jellyfish. And there's opsin vision genes or light responsive genes adjacent to these stinging cells, and they use that to modulate their stinging. This video might work. Oh yeah, it's on my laptop. We got it there. So again, this is octopus skin. We took a little piece of octopus skin, put it under a microscope, shined a light on it, and they have this camouflage, but just from this divorced skin from the rest of their body, shine this light, and it causes this response of the these chromatophores. And this is the response curve. Different wavelengths of light is consistent with this opsin gene, and we find the opsin gene expressed in the skin of octopus as well. So that's trauma. Third origin story is destiny. I almost said density, like Back to the Future, but okay, so that's me as a little kid, Green Bay Packers fan, but I used to always collect fireflies in my backyard and just fascinated by this complex system of producing light, and they use it as a communication signal. And so learning more about that, and this kind of feels like my destiny to study bioluminescence. This is a fish. The fish itself is not bioluminescent, but it's biting a small crustacean that you can kind of see swimming past. The small crustacean is now what I study. They spit out this whole cloud of glowing mucus, and it startles the fish, or maybe it's distasteful to the fish. That's also traumatic, I would say, but we're in the density section. Getting eaten, I imagine, is traumatic. So this is the origin of many bioluminescent systems, is in these aposomatic signals or anti-predator signals. They startle or warn the predator. But in addition to that, like the fireflies and even underwater, some animals use bioluminescence for courtship signals, and that's what's going on here. And this is what we study in the Caribbean lately. And bioluminescence originated at least 90 times. So we reviewed all the literature, again, mapping the close relatives, which ones have bioluminescence, which ones don't. And this is at minimum 90 times bioluminescence originated. So again, it almost feels like, I keep saying density, almost feels like destiny. We are maybe destined to become bioluminescent one day. I don't know. But we took advantage of that replication or those separate origins and ask, well, is there something that we can learn generally from bioluminescence? And we looked in particular at the groups that have bioluminescent courtship. So like the fireflies that you may know about, or the ostracods that I showed. And we compared groups with bioluminescent courtship and counted the number of species and compared it to their closest relatives and counted that number of species. We wanted to know if bioluminescent courtship leads to more species. And in 10 out of 10 cases, we found that to be true. That in the cases where bioluminescent courtship has originated during evolution, it leads to more species. And so this has been kind of debated for a long time, whether sexual selection, choosing mates based on some display or competing for mates based on some display, leads to more species. And this is one of the best pieces of evidence that we have found from studying bioluminescence. And then I'll talk a little bit more about just some of the most recent work that I'm working on now. Is I'm interested in the molecular basis of the diversification of these courtship signals. And so this is some fun photochemistry here. These are producing light with chemistry, spitting out these bioluminescent mucus dots in patterns to attract females. And everywhere we go across the Caribbean, there's different species. There's dozens and dozens of species. We go 500 meters from where we were before, and we find a whole set of new species. And so one goal of mine is to begin to quantify and catalog those species. This is what the animal looks like. It's about the size of a sesame seed producing these lights. And working with undergraduate engineering students, we're building underwater cameras that create 3D patterned videos of these bioluminescent courtship displays, and that we call WALL-E, waterborne autonomous low light electrostereo videography, because it kind of looks like WALL-E. And then we're finding these collective signals in the ocean where they respond to each other. So I'll just let you watch this. Hopefully it works. Oh, it looks like it's not working anyways. Oh, there it goes. No. Well, I have one more. So these are a brand new species we discovered in Panama, and we call it NOL. We always give them codes, field codes. NOL, it reminded me of Christmas lights. And if we plot the light over time, they're basically like these synchronous fireflies, but one of the first times of finding this in the ocean. And then I just wanted to mention very briefly, machine learning has come up a lot, and we are using machine learning to predict the phenotypes of these genes, of these signaling systems from the genes themselves, predict the color sensitivity. So this is the opsin genes themselves, and we can do a pretty good job of predicting what the gene is sensitive to, what color. So told 3 stories today: origin stories, chance, trauma, and destiny. And so maybe the take-home message is that just like our own personal histories, just like the history of humans, deep evolutionary history shares the similar kind of thing, that strongly influenced by chance, trauma, as well as destiny. And maybe my parting question: if the deep origins of light responsiveness are rooted in stress, does that change how you think about the role of light in life? So that was kind of one thought process of the role of stress in light responses. And then I'm also interested in the bioluminescence either way. And I'll stop there. And I went a little over time. I apologize for that. Thank you. Yeah.
I love your comment about light as toxicity and thinking as origin of light sensors to find out the toxicity, because as someone who's worked on more, very high resolution type of visual systems, I always just think about visual as information. So I just want to highlight that, because it goes into something that I'm thinking a lot of. You have a laser pointer or a strong laser and you burn a hole in the wall, right? But mostly we think of light something totally benign and has no influence on us. And so where is the balance is one of the things. So I just wanted to bring this up. But I do have an actual question too, and that's my naivety too. But this whole bioluminescence thing, so this is mostly bacteria-based. Is that correct? And is it the similar actual chemical mechanism as you would find in the firefly that you find in the bioluminescence of the ocean?
Yeah. So many of the origins of bioluminescence are the symbiotic relationships with the bacteria, and the bacteria produce the light. So that's what you're alluding to. These ostracods and all the ones that use bioluminescence for courtship, the animal themselves creates the genes for bioluminescence. The question of whether these sea fireflies, the chemistry is similar to the fireflies that you know of. The small molecule, which is called the luciferin, is a different molecule. The proteins are totally different and non-homologous, but the similarity is that all of these bioluminescent reactions are oxygenation of the small molecule, but there's about 8 different known small molecules, and then every one of those 90 plus origins of bioluminescence is a different protein.
Wow.
So yeah. Well, yeah. Well, no, it has to be, yeah. And then a lot of times they share this small molecule they have to eat their prey to get the small molecule to bioluminesce like a vitamin. So there's some fish that eat these ostracods. The fish can't bioluminesce until they eat the fish. I mean, eat the ostracod. Yeah. Yeah.
What a great presentation. Kind of speculatively and kind of naively again, we talked about the 90 different origin stories for these processes. And
Yeah.
maybe there's some part of destiny where we're all moving towards the bioluminescence.
Yeah.
What,
That was a little tongue-in-cheek.
I know, but what are moments when we recognize these origin stories and the destiny? What is the destiny of the bioluminescence in these species?
Well, yeah, I mean, the way I think about destiny is more about in terms of predictability. As an evolutionist, could we use commonalities to be able to predict what would happen? And so one example that we're just working on now is multiple of these different bioluminescence systems that they need to be able to store the small molecule in a safe way. And one way to do that is a chemical process, enzymatic process called sulfation. You just add a sulfate group to it. But there's basically only one way to do that we know of in all of biology. So all of these separate instances have used the same process called sulfation, the same gene family called sulfotransferases. And to me, that's what I'm thinking more about in terms of destiny, is that we could predict that, and we did, because there's only, as far as we know, one solution. So it's a little bit
There are
complicated.
discrete moments within a similar trajectory.
Absolutely, yeah. Parts of it are predictable, parts of it are chance, and parts are drama too, right? Because they're getting eaten, right? So, yeah. And yeah, that's the history of life that fascinates me. Right, thank you. Oh, yeah.
Very interesting talk. I was curious on how do you measure the light intensity of fireflies?
Yeah.
Because they are moving around. Is that like the overall light intensity for a known number of fireflies?
Yeah, there's different ways of measuring the light intensity. So we're using those cameras for one, and we can get fairly crude measurements, like not spectral measurements. Then you can use spectroradiometers that are highly sensitive. So Morgan brought one by our lab, and we use that to get the spectrum, or we can use a spectroradiometer if we have the isolated animal. And then a great thing about bioluminescence is we can take the protein itself and clone it into cells outside the animal, and then add that small molecule substrate and measure the phenotype. So that's where I was kind of rushed at the end, but that's where we're trying to get to the connections between the genotype and phenotype, the connection of the color of and the kind of kinetic reaction of the light production. Yeah. All right, thank you. Yeah, that is on the computer now. That's good. That I had mine on there. That was my drive. Oh, how do you get to the?
Very briefly do end stream and start stream.
How do you get to the?
I don't know, this is PC.
Do you see yours?
Mine is down there.
Oh yeah.
There it is.
Perfect. Next speaker is Professor Jan-Erik Nilsson from Lund University. Some of you heard him give one talk yesterday, and today we'll talk about motor vision.
Thank you, Elke. So what I'm going to talk about today is kind of related to what I spoke about yesterday, but it's still kind of another dimension of it. So we're all affected by light, obviously, but not just the amount of light. We're affected by the visual environment that we have around us. Clearly, everyone knows that and feels that, but we know very little about it. If you look at these different environments, they are very different environments. If you would find yourself in any of these environments, you would get a kind of a different feeling about it and possibly want to do different things and get in a different mood. But the way this has been dealt with before is that we've measured the amount of light. People have used irradiance or illuminance meters, lux meters, basically, which tells you how much light there is that falls onto the environment. If you use the lux meter here, you will, in these four environments, you will actually get rather similar readings. And so if you just try to treat light as if it was a temperature, you will get the wrong answer. There is not just one light intensity here, because if it was, we would not see anything at all. So the fact that we see something is because light actually varies, and it varies a lot. Within a single scene, it's about a difference of 100 times between the dimmest and the brightest parts of a typical natural scene. And that's a lot. Animals, of course, also use it for finding the place to be and decide what to do. So the animals cannot just measure the amount of light. They have to use information from the image. The spectral content, the different colors, where light comes from, all that will have to go into the judgment of a habitat. So this affects behavioral states, as we call it in animals. We would call it states of mind or something like that in humans, but it's all the same thing, which then, of course, there are lots of things that influence the behavioral state. But what has not been very much explored is how the visual system, not just measuring the amount of light, but the visual image, how that actually affects our state of mind or the behavioral state of animals. Where does this happen in the brain? I can talk for hours on where it happens in different types of animal groups, but among vertebrates, or in particular in mammals, it's parts of the brain which we don't use for normal vision. It's other specific, much, much smaller parts of the brain that does this. But these parts of the brain were previously thought just to acquire information about the intensity. Now it turns out in all of those parts of the brain, there is actually spatial information. There is an image there. So that means that when we measure light, to use it to understand how we experience the world around us, then we can't just measure illuminance. We have to measure light in a slightly more complex way. And there are actually many other problems with the kind of light measurements that people have been using so far. For the first, of course, we have to measure light from the perspective of the animal or the human, from where they are. We can't look at the amount of light falling onto the environment, because then we get this kind of temperature measurement, the average, which doesn't really say an awful lot at all. It's an extremely ambiguous signal, because the amount of light can vary by so many different factors that we don't really know much at all about it. So we need more information. So obviously from the eye's perspective is important. And we shouldn't just measure light as energy, which engineers like to do. Because no animals, humans included, measure light as energy. We measure light, we count photons. And if you go to different spectral regions, then you get it wrong if you actually measure energy. So you have to measure photons. You don't make a tremendous mistake if you measure energy, but you do generate an error by it. And then other important things are that humans and animals, we have very coarse spectral sampling, different colors. We pick them up in 3 points. Spectrometers can divide it into nanometers. Animal eyes and human eyes divide the spectra into 3-4 different large chunks, and many mammals actually just into 2. So spectral sampling is coarse in animals, although spatial sampling, where light comes from, that is much finer, even though it doesn't need to be super fine. It is usually much finer than hyperspectral cameras and any other kind of light measurement gear can provide. So we need fine spatial sampling, at least much better than what we normally use, and we need to include elevation angles, because that is what actually differs. In that direction, in the azimuth direction, it's kind of random, depending on where you are and what direction you're pointing. But what signifies what is typical for an environment is how light varies in the vertical angle. No change, yeah. So the way we do this is we actually use a consumer camera, put on a fisheye lens, which is 180 degree wide, and we put a bubble level on the camera, so we always have it horizontally oriented, which means that the center of the middle plane through the image, that's the horizontal plane. We've got information from straight up to straight down, and then cover half the visual world. If we want to cover the entire visual world, just take another measurement in the opposite direction, and you've got the full sphere. So we measure light from all directions. And then we remap this image, so we get it in pure elevation angles in one direction and pure azimuth angles in the other. And actually, that image you see here, that is one of those, that is actually a circular image initially, which has been made square. So we've got pure elevation angles there and azimuth angles there. Now, it's not a single scene, because we take many scenes in an environment and add them to one another, and then we extract data from that pile of images. So we can extract information about how the intensity profile goes from straight down to straight up, how the amount of visible structure, how much light varies, which is another important thing, and the spectral oops. Well, actually, you can use this one. How light is spectrally composed, or how composed of different spectral bands, how that varies with elevation angle. I'm just repeating very shortly what I said yesterday. We can, using this, discriminate very, very clearly between different types of environments. We can discriminate between different weather conditions. This is actually a single scene from La Jolla Beach, San Diego, and starting with very nice weather and going worse and worse until fully overcast. And you can see how you can actually know from these curves, you know exactly what on the way from fully clear weather to fully clear skies to full overcast. It's easy to see that. You can also look at different seasons. For example, this is not a single scene, it's a complete environment, sampled once a month over a full year under the same weather conditions, same time of day. And you can see how dramatically the curves shift over the season. So it's very easy to judge what season there is from these vertical light gradients. And you don't need very many points on these curves, as you see. So we know that from these vertical light gradients, you can extract biologically very important information about where you are, what time of day it is, what season it is, what weather condition it is. And if you're an aquatic animal living underwater, then you can also work out how deep you are. And you can work out these things independently. They are no longer confused into a single measurement. You can work out all these things separately. So now I'm just going to say something about how you can apply these to all sorts of interesting things. These are measurements from environments where chickens are bred. Oops. Let's see there. So that's a chicken house. You can see what it looks like roughly in there. You can see the curves there. And then this chicken house, they were exceptionally proud of the fact that they had introduced ultraviolet LEDs in their chicken house. So the chickens would get ultraviolet, which was supposed to be much more natural. So we went out and measured light in the natural habitat of red jungle fowl, which are the original species that we have now. Of course, bred to death almost. So they may not be very natural anymore. But anyway, we looked at the natural environment for red jungle fowl, and there is basically nothing that is the same. This is a logarithmic scale here. So you see 16 to 17 there, the chickens actually get 13 to 14. It's about 1,000 times dimmer than they should have. They usually have a dark horizon. Now in captivity, they get a bright horizon. The amount of visible structure is way larger in the natural habitat than in captivity. And they were so proud of the amount of UV light, but they're overdoing it massively. The violet things here, that's the amount of UV light in natural habitats. It's not that much, but it is there. And for other chicken houses, they don't have any UV light at all. But the ones that now decided to do it, they massively overdid it. So it's about 100 times or 10 times more UV light than they're supposed to be. I think that you could keep these chickens much more happy and bring up productivity if you make the lighting conditions more natural. These are typical human light environments. And in this case, I've just selected the spectral curves from red, green, and blue. So here you've got typical, that's a cafe, that's an art museum, and that's a classroom. They all look pretty similar. Actually, all indoor environments look pretty similar, but they don't look like no natural environments. These are 3 natural environments: a savanna, public garden, and alpine meadow. They are different, but they are also distinctly different from the indoor environment. So possibly we expose ourselves to rather unnatural environments all the time, and maybe we would be a bit happier with slightly more natural environments. And now with modern LED lighting, that is actually, we can easily accomplish that. We can make something like which with diffuse light illuminating the ceiling. We can provide exactly these curves with rather limited or rather simple ways. Todd, you pointed out that we are destined to become bioluminescent. We're already emitting light through our technology. Yeah. And so we're polluting the light pollution is a bad thing. And it's not very sensitive, this thing. You have to keep it, for people speaking later, keep this slide changer close to in that direction, otherwise it won't work. Here we have a scale from these are measurements, a single scene from an Australian desert, a place where there is absolutely one of the few places remaining on this planet which is completely free of light pollution. So that's the middle of the night, that's late dusk, sunset, and noon. So that's the daily variation. You can see what the place looks like. And this is on a single scale, just the intensity curves, which go from the purple curves. And I've introduced a few other curves here from light polluted areas that are differently light polluted. There we've got 4 kilometers from a remote resort. So that's really not very much light pollution, but that is starlight, and 2 log units higher than that, roughly where you have that other dashed curve, that's moonlight, full moonlight. And so this 4 kilometers from the remote resort almost takes the intensity halfway towards moonlight and erases that natural dynamic between starlight and moonlight, which many animals, including humans, I think, are somewhat dependent on. If we get a bit further in, that is 1 kilometer from a city border, then we're definitely above moonlight. Suburban street at night, way above even mid-dusk. So we erase all that natural dynamics. It's gone. And not just for humans, for animals too. And animals that are night active, they have lost their night. And they start to behave in ridiculous ways. And I'll give you a short little interesting story from South Africa. They had problems on a football field. The football field was covered with large dung beetles. And they were stumbling over the dung beetles, but they are massive. These are fantastic dung beetles. It wasn't that the football field smelled of shit. That was not the problem. The dung, because all the dung pads, they were in the farms around. They were not there. But why did all the dung beetles go to the football field? It turned out these are nocturnal, night-active dung beetles. They flew in there, and when they got into the football field, which was lit by really strong lights, but of course a black sky, fairly black sky above, they judge what is up and down using their eyes. So they took off to fly, and they realized it was much brighter down there, darker there. So that must be up, that must be down. They turn around and crash. They take off, turn around and crash. Take off, turn around and crash. So as soon as a dung beetle flew over that field, they got trapped and they couldn't get away. So they collected all the dung beetles from kilometers around. They were all in that little football field. Now, finally, I'm going to say something about something that was a bit of a surprise when we started to do these things. When you start to add different images on top of another, so you remove the scene-specific information largely, but you can't stack too many images such that you get a pure intensity gradient and color gradient. You need to see some of the structures, a moderate number of scenes on top of another, and then you get something that looks very painting-like. This is one example. I'll show you a few more. They look sometimes a bit like impressionist paintings. So I think it says something about the way we experience our world. And structure, visible structure is important. It's really important. This is another, I think I've got the last one here. So they really give you an impression of being in a particular place. Yeah, they are. This is a fantastic, cheap way of producing nice art.
No AI required.
Exactly. None. Right. Thank you for your attention. Yes.
I think you raised a really crucial issue. Let me ask you a question. Is there, I mean, I work on mammals, so this question isn't so obvious for people who work on mammals, but there must be good evidence that some species decode Raman dust spectral composition.
Yeah.
Can you tell me about that?
There is very little known about it. I mean, it's obvious now from our measurements, it's easy to see the information is there.
Yeah.
Now we know exactly what animals ought to pick up, and it's very little information. And that is what makes it so hard to investigate, because you need low resolution. You don't need very many points. So you don't need a very large number of neurons in your visual system to do it. So a small number of neurons. Another thing is that those neurons have to be slow. Slow neurons have thin neurites. So a small number of neurons that are thin, it's not going to be a massive part of the visual system. So even though this part of the visual system is really essential and very important, it's going to be tiny. And there are such tiny parts, and especially in the vertebrate system, that you've got these light-sensitive retinal ganglion cells. They project to various different areas, and many of them project to tiny areas. Some set our biological clock, others set our mood and things like that. So those are the bits we have to look at, but it's yet rather unexplored. Yes?
Just spectral graphs, super interesting. But something that I noticed is that the far red, and also the long wavelengths and the short wavelengths graphs, often in the natural scenes in the horizon, they shift dramatically, where the green ones,
Yeah.
the most steady ones.
Yeah.
And of course, it occurs to me that green is also the photoreceptor for our thoughts, and also almost every animal, if it has any photoreceptor, green seems like the standard which most animals have. Is there a connection? Is this
Well,
coincidence, or-
even if you look at the green curve, it often makes a little kink at the horizon. So the horizon is always important. But then there is generally much more blue light above the horizon and very little blue light below the horizon. And the reason for that is that most objects they're not very good at reflecting blue. They're even worse at reflecting ultraviolet. So the ultraviolet is really dark below the horizon. The blue world is fairly dim below the horizon. When you get up to the green world, then yeah, it starts to balance out a bit.
But the red one was pretty.
Yeah, and the reason for that is that the sky, especially when you've got a clear sky, the blue sky doesn't contain very much red at all. Direct sunlight does. So you get direct sunlight shining onto the ground, which contains lots of stuff that is pretty good at reflecting red light. So you get lots of red light from below the horizon, but the general intensity when you look up, not right into the, that's actually another problem. We always measure median intensities, not average intensities. Average intensities will lead you wrong because it'll give way too much bias to strong light sources. Our eye or animal eyes, they are usually saturated, those few pixels that look at the light sources anyway. So they don't contribute to our general impression. Right?
All right, thank you. We're just making a quick stop and start.